Hi, my name is Lorena, and I'm a freshman working at the Pezzulo lab. My talk today is on the project that I've been working on for the past couple of months, and it is called Ascaris worms secrete a human periostin-like protein that may facilitate airway invasion. So it is known that parasitic nematodes cause human disease. Among these nematodes are Ascaris lumbricoides, which occupies the human lungs and induces inflammation and asthma, and also Ascaris suum, which live inside pigs and cause similar effects. And pig infection does lead to major economic losses to the pork industry. Ascaris infection begins with ingestion of the eggs, which is usually from contaminated drinking water. The eggs hatch into larvae in the GI tract, and then the larvae travel through the blood vessels and the liver into the blood vessels of the lungs. However, migration from the blood vessels to the airway lumen can cause airway damage, which could alert the host immune system to kill the worms or could also injure the host. So the worms need a way to travel to the lumen and infect the lungs without harming themselves or the host. And to do this, parasites often mimic host functions to camouflage themselves from the immune system. So when there is human airway damage, airway cells secrete molecules that facilitate cell migration and extracellular matrix remodeling. An important example of one of these airway remodeling proteins is periostin, which is secreted in large amounts by airway epithelia after lung injury and inflammation. So we hypothesized that Ascaris secretes a human periostin-like protein to avoid alerting immune cells and facilitate its migration into the airway lumen. My first question was, does the Ascaris suum genome contain a human periostin homologue? So to investigate this, I took the human periostin amino acid sequence and I blasted it against the Ascaris suum genome. And I found that there were 2 hits of proteins in Ascaris that were similar to human periostin. This table shows the gene name, the length, and the percent ID, which is how similar the protein is to human periostin. From this, we can conclude that human periostin matches 2 genes found in Ascaris suum isolates. So my second question was, does Ascaris extract contain a periostin-like protein? And to investigate this, I used Ascaris extract, a powder containing dried Ascaris worms, and I measured the amount of periostin in various concentrations of the Ascaris extract using an ELISA for human periostin. My results are here on this graph. So on the x-axis is the concentration of Ascaris extract, and on the y-axis is the concentration of periostin. And as you can see, the concentration of periostin increases as the Ascaris extract concentration increases. And from this, I concluded that Ascaris extract contains a periostin-like protein. However, Ascaris extract is commercially available and it is unclear how it's prepared. So we cannot conclude that Ascaris actually contain a periostin-like protein from this data. So this leads to my third question, which was, do living Ascaris secrete or contain a periostin-like protein? To look into this, we obtained live Ascaris worms from an Iowa meatpacking plant in collaboration with Dr. Martin at Iowa State. And we maintained the worms in a buffer at 33 degrees Celsius. So there were 2 fluids that I collected from the Ascaris. First, I collected Excretory and Secretory Products, which act as an immunosuppressant on the host. And second, I collected Perienteric fluid, which is highly allergenic. And I collected the Excretory and Secretory products by collecting samples of the buffer where the worms were kept. And I also collected Perienteric fluid upon dissection of the Ascaris. So what I found when I analyzed the fluids with a periostin ELISA was that both fluids contain a periostin-like protein. So here you can see the left bar is perienteric fluid and the right bar is the buffer containing excretory and secretory products. And this is measuring the amount of periostin. So it's important to note that we measured the excretory and secretory products in the buffer containing the worms. So it was probably diluted at least 1 to 1,000. So the bar on the right would likely be a lot higher if the ESP had not been diluted so much in buffer. So from this, we concluded that Ascaris perienteric fluid contains a periostin-like molecule. And also Ascaris excretory and secretory products likely contain a very high concentration of the periostin-like molecule. So an overall conclusion that we can make at this stage in the project is that Ascaris suum secretes a periostin-like protein, which may facilitate its migration into the airways. And also Ascaris and human airway cells production of the similar proteins could be an example of convergent evolution. So in the same extracellular matrix environment, both the cells and the Ascaris evolved to make this protein, the periostin or periostin-like protein, so that they can move around in this extracellular matrix environment. So our next steps in the project are to clone the periostin-like protein and then expose human airway fibroblasts and epithelia to this recombinant protein and analyze their response. So here are my acknowledgements. I would like to thank everyone in our lab, Michael Catlin and Joseph Sabner, and Richard Martin for their help with this project. And thank you all for watching.